Hi everybody, I am Emre from Istanbul University. The title of the speech is Trichoderma atroviride triggers alterations at epigenetics, transcripts, oxidative stress, and apoptosis levels on Fusarium graminarum. Fusarium graminarum, here we see the 7-day-old cultures, Mycelia and spores, is a species complex, more than 15 members, has a hemibiotrophic life cycle, asexual and sexual reproduction, infects several plant species, including all grain smell cereals. Primary pathogen of Fusarium head blight and crown rot diseases in humid and semi-humid regions. The genome data of the Fusarium graminarum is released. Fusarium graminarum is the casual agent of Fusarium head blight and crown root rot diseases worldwide, and especially Fusarium head blight is one of the most important destructive diseases of all small grain cereals worldwide. The epidemics led to the yield losses, reduction in crop quality and quantity, and mycotoxin contaminations. A struggle with Fusarium graminarum and its diseases includes several different strategies such as antifungal treatment, resistant cultivar development, genetic engineering approaches, and essential oil compound treatments. But here we describe the novel strategy with the molecular details. Current study provides data on relationship between Trichoderma atroviride and Fusarium graminarum at epigenetics, transcript, oxidative stress, and apoptosis levels. In Meta-Metas, firstly, we would like to introduce our Fusarium graminarum and Trichoderma atroviride strain PH1 and TR8. All fungal samples were grown on potato dextrose agar for 7 days, and mono and dual cultures were maintained as control and experiment sets respectively. And agar deletion techniques was used in order to obtain linear growth rates. In growth and cytotoxicity assays, a common protocol for WSD1 test was used in order to obtain cell proliferation assays by colorimetric tests. Here we see the wavelengths for spectrophotometer tests in control and experiment tests. Genomic DNA was obtained by using a common procedure from a sodium dodeosulfide based protocol. Quality and quantity of genomic DNAs were checked by agarose and spectrophotometers. Random amplified polymeric DNA test, a common protocol was followed. Here we see the details, and genomic template stability and resolution power values were recorded. In CREDRA assays, we have obtained methylation as polymorphism percent by using Neal and Lee's coefficient, and OPGAMMA derivative analysis were also used in genetic and epigenetic diversity evaluation. And total RNAs were extracted by using 3P reagent. The DNA treatment was also carried out. Agarose and spectrophotometer assays were used in order to check the quality and quantity of total RNA molecules. A commercial kit was used in order to obtain cDNA molecules, and gene expression assays were conducted on CyberGreen1 fluorescent dye and Quantidio5 system. Fold changes in gene expression were evaluated by Livak and Schmisken formula, and beta-tubulin was endogenous gene. Catalase, MST20, and TRI4 genes were used as target genes. In fluorescent microscopy assays, apoptosis-like process was evaluated by Akron orange ethylene bromide dual staining, and oxidative stress was investigated by ECFDA staining. And also 2Ks LCD test and 1-way ANOVA test were used in statistical tests. Results, here we see up to 50% decrease in linear growth rate was obtained by agar deletion technique, and also WSD1 test. Up to the 50% decrease in linear growth rate was obtained, and cell proliferation. In genomic template stability and methylation analysis, genomic template stability was affected by Trichoderma atroviride treatment in Fusarium graminarum as a result of 66 26% decrease in genomic template stability. Similarly, 26 and 13.88% methylation values were recorded. Also, type 2 and type 3 methylation alterations were recorded due to Trichoderma atruviridae treatment in Fusarium graminearum. In relatively low level of RP values were recorded. Here we see RP values as a low recorded. Idiomorphic and polymorphic bonds were also recorded from rapid analysis. And here we see the methylation status of the MSP1 and HAP2 digestion profiles. In gene expression assays, scientifically acceptable E values and Melton scores were obtained, and downregulation of TRI4 gene and upregulation in catalase and MST20 genes were detected. Here we see the heat map and also fold changes in gene expression with this scientifically acceptable and scientific difference values for fold changes in the gene expression. Also, presence of apoptosis-like process. Here we see in experiment sense in D section and also H section, we can see oxidative stress process were present in experiment cells. Management of Fusarium head blight and crown rot includes several approaches such as grass-resistant cultivar development, fungicide treatment, plant microorganism, their essential oil treatment, and antagonistic microorganism usage. Fungicide usage has been most frequently preferred in fields. However, resistant development to the common fungicides such as thiophanate methyl and carbendazim or another tebuconazole, tetraconazole have been increasingly reported from literature. And antagonistic microorganism usage has several advantages in comparison to other strategies, being cost-effective, revealing data within a short time period, and non-laborious. The most of the studies in Trichoderma sp and Fusarium sp are conducted on Fusarium oxysporum and Trichoderma harzanium, but in the study we have shown that Trichoderma atruviridae could lead to the physiological alterations in Fusarium graminearum by different levels, including epigenetics and genetics, and also transcript levels changes. And Trichoderma atruviridae may be accepted as potential secondary metabolite biosynthetic blocker in Fusarium graminearum, but further studies could be conducted on transposon mobility and implanted tests. Thus we can help in obtaining useful, revealing more comprehensive data related to the antagonistic effects of Trichoderma atruviridae or related species on Fusarium graminearum, or maybe another plant important, or maybe other top 10 phytopathogenic fungus and or close related phytopathogenic fungi. Thank you for listening me. Please see www.phytopathology.com. You can download this speech PDF file by this internet service.